Okay, so I'm going to be talking about the research I conducted over the past 5 years here at Utah State University. This has primarily looked at how colors of light affect plant growth and development, specifically looking at far-red and phytochrome. So my title, Phytochrome Physiology and Plant Perception of Far-red Photons. There's a lot of jargon in this title, so I want to try to kind of demystify it a little bit. So first, phytochrome. Phytochrome is a photoreceptor in plants that, and a photoreceptor is a protein within plants that has a component that absorbs light, which is called a pigment. So when the pigment part of the protein absorbs light, it changes the shape of the whole protein, activating the protein into a form that can regulate downstream processes, often through manipulation of gene expression, but also through interaction with cell membranes. So phytochrome is one such photoreceptor. I'm also going to talk a little bit about cryptochrome. Then the other term that's probably unfamiliar to a general audience is this term far-red in the title. Far-red is a very plant-specific term. So when we learn about light in elementary and middle school, we learn Roy G. Biv, red, orange, yellow, green, blue, indigo, violet. And these colors are useful for the human eye. We're very sensitive to these different types of colors, but plants don't necessarily see light in the same way. Here's a good point to say that I'll be using the word light and photons fairly interchangeably throughout this talk, but photons is really the more appropriate term. So if we look at red here, far red is just kind of farther on in this direction, and our eyes aren't too sensitive to it. It appears as dim red light to us, but it has big effects on plant growth.
That turned on.
It is turned on. So it's useful to talk about broader categories in plant physiology. So even though there are these narrow bandwidths, we find it useful in horticulture to broaden this out into these 5 useful categories: ultraviolet, blue, green, red, and far red. So in my talk today, I'm going to be talking about one study I did on blue and green, and the rest are primarily going to focus on this far red region. I've done a little bit of work on ultraviolet while I've been here, but I'm not going to talk about that. So these broad categories are fairly coarse, but they're also useful. And we might be able to split them up into slightly narrower bandwidths. For example, I put ultraviolet here as 300 to 400 nanometers, but really we usually talk about UVA and UVB. And the divide for that is somewhere around 315 to 320 nanometers. And it depends on the paper you're reading for where exactly that divide is. And likewise, we think it might be useful to split far red up into long far red and short far red, with short far red probably having more potent effects on morphology than this longer far red. Before moving on, I want to point out that we're looking at the electromagnetic spectrum here, and we're looking at just the visible portion of it. And this graph with this exact x-axis is going to appear throughout this presentation. So it's really useful to kind of get your head around wavelength. So you're increasing energy of the photon as you move to a shorter wavelength, and just kind of get an idea of the different colors associated with these wavelengths. The next set of definitions I want to go over cover what we consider photosynthetic photons. So traditionally, for the past 50 years, we've considered photons in this region from 400 to 700 as photosynthetic photons. And if a photon lies outside of this range, it's not considered photosynthetic. So we call this photosynthetically active radiation, or PAR. So a photon at 399 or 701 has not been considered photosynthetic. And it does so happen that these photons are the same that we use, that we can see. If you count the number of photons within this region incident on a surface, that's called photosynthetic photon flux density, or PPFD for short. So this has units of micromoles per meter squared per second. it's basically the number of photons hitting a unit area per unit time. And it's pretty synonymous with intensity. I might use the two words interchangeably in this talk. But as PPFD increases, your photosynthesis increases. But recent work out of this lab and one of my committee members' labs, Mark Van Ursel at UG, have been doing work. This is primarily done by Shuyang Zhen, showing that this region should probably be expanded out to 750 nanometers. These photons from 700 to 750 also appear to be photosynthetic. So we've expanded this definition. We've said extended photosynthetically active radiation and stuck a little E in front of this. So when we do these studies in horticulture, looking at how colors affect especially plant shape, it's really useful to keep your photosynthetic photons equal, because otherwise changes in growth might be due to changes in photosynthesis. And if we're really interested in morphology, we want to keep this region equal and just change the ratios of the colors in the spectrum. Because a lot of my studies involve far red, I kept ePPFD constant and just manipulated the ratios of the colors. So I'm going to talk about these two guys later in the presentation, but at this point, I wanted to talk about how plant photobiology was done largely in the 20th century and recent developments in technology that have changed the way we are able to conduct experiments. So in this image here, they're using a prism, same type of prism that Isaac Newton used to split up white light into its component colors, what I was showing you on the previous two slides. And basically they just placed plants in this at different wavelengths and saw how it affected things like germination or stem elongation. So you could either use prisms like this to investigate effects on plant morphology, or you could use filters like a blue glass, which allows blue penetration through the glass and filters out green and red wavelengths. Both of these techniques were useful to figure out how colors of light affected plant growth throughout the 20th century, and we learned a lot of useful things. But now we have LEDs, which are a much better tool to conduct experiments. So you can see the narrow bandwidth of different types of LEDs in this image here. And we can mix and match these together, apply them at different intensities to create really careful spectral treatments in order to investigate how different wavelengths are affecting plant growth. In addition to their use as a research tool, LEDs are also really useful for industrial applications. So this figure actually comes from one of these two papers I've written. And if you take the area under the curve, it looks at the, it basically is you're able to compare the efficiency of these different types of common LEDs. And these two papers are looking at how efficient LEDs are compared to other technology, how efficient they are compared to each other. And it does turn out that under the right conditions, LEDs are twice the efficiency of the next best lighting technology. So we wrote these papers looking at comparing efficiency and looking at efficiency losses when you take a single LED and incorporate it into a whole lighting fixture. And we wrote them with our colleague, Morgan Pattison. He is a LED scientist. He consults the Department of Energy. And he actually got his PhD with Shuji Nakamura, a Nobel Prize-winning scientist for development of a high-efficiency blue LED. There are two funding agencies that have funded my research over the past 5 years, and they're really interested in this efficiency question of LEDs. One is a USDA project called LAMP. LAMP stands for Lighting Approaches to Maximize Profits, and it's interested in the economics of lighting in greenhouses and like vertical farm settings. And the other project is a NASA project called CUBES. CUBE stands for the Center for the Utilization of Biological Engineering in Space. And this project is interested in using biological means to create a sustainable environment for a few astronauts on Mars. So both of these projects are really interested in how efficient different lighting technologies are and how efficient they can be. So these two papers are useful for those projects. But they're not only interested in efficiency, they're also interested in the photobiology I've been working on. So on that note, when we think about how colors of light affect plants, it's useful to think about the evolutionary pressures that have driven the evolution of these photoreceptors. So if we take a measurement of the spectral trace of full sunlight, it'll look something like this. So this is the same x-axis I've been showing you in all the previous slides. So again, this is UV ultraviolet, blue, green, red, and far red. And the orange line just shows the intensity of the photons at each wavelength. So if we grow a plant out in the middle of the field, my cartoon image, it might look something like this. But now if we take a second measurement and take it underneath the shade of this tree, now the trace will look something like this. And you can see it's highly enriched in far red, and due to the photosynthesis of the leaves, this whole PAR region gets significantly reduced. So now if you take the same plant that was growing out in the middle of the field and grow it underneath the shade of this tree, two things can potentially happen, and it depends on the species. First, you could have a shade avoidance species, and this is typically displays elongated stems and a decrease in leaf area. And the other thing that can happen is you can have a shade tolerant species. Shade tolerant species typically will increase their leaf area and won't get much taller. And here's a good point to mention that a lot of my research has looked at how these colors of light affect this parameter of leaf area. And if you think of a leaf similar to a solar panel, the more area of solar panels the larger area you're covering with solar panels, the more light you can harvest from the sun for use in electricity. And similarly, if a plant has a wider leaf area, it can capture more photons for use in photosynthesis. And because of this, leaf area is highly correlated with biomass accumulation and yield. So from an applied standpoint, we're really interested in leaf area to increase yields. This response to plants in the shade was observed way back in the 1700s. So I have a quote here from Stephen Hales. He was a clergyman who was interested in the natural environment. He actually contributed to the discovery of photosynthesis by finding out that leaves could exchange air with the air could be exchanged between in and out of leaves. But this is his quote: "Beans and other plants which stand where they are much shaded, being thereby kept continually moist, do grow to unusual heights and are drawn up, as they call it, by the overshadowing trees, their parts being kept long, soft, and ductile." So this is a quote from several centuries ago, basically mentioning that this shade avoidance morphology occurs when you put a plant in the shade. And although he did observe this, really it was the late 1960s and early 1970s that we started realizing that these shifts in spectrum induce these changes. Before I get into this big spike in far red, I want to talk about blue and green. So if we look at the percent blue and percent green as a percent of this photosynthetically active radiation, blue is about 27% at full sunlight and green is about 35%. Now, if we compare that to the percents in our shade measurement, those are, we can see that the blue goes down a little bit and that the green increases. The total effect that this will have depends on how deep of shade you're in, but we do see that blue decreases and green increases. And this is somewhat obvious when you think about it in the fact that leaves are green. We view leaves as green because they're using slightly less, they're absorbing slightly less of those green photons. Now, as I mentioned, plants do use 400 to 700, all these photons for photosynthesis, but they absorb blue and green really well, while green is has a slightly higher transmission, and it's slightly enriched in the shade. So this could potentially be used as a shade signal. When we think about how light affects plant growth and development, it's useful to think about what the underlying response is without any stimulus. So these are actually some demonstrations I brought to Washington, D.C. When I went to go talk to some Congress people about the NASA project we were working on to try to get them to give more funding to NASA. And this was a demo I brought to show how different colors affected plant growth. And what you can see here with these dark plants is that those are the most elongated of all three of these. And so what this means is when plants are grown in the dark, they will continue to elongate. And if you think of a seed in the soil, it'll continue to elongate until it reaches full sunlight. But when we apply blue light, the plants stay fairly short, and then green is in between. So it's important to keep in mind here that the actual stimulus, which is the blue light, inhibits elongation rather than promoting something. So when we think, or when we look at the photoreceptor responsible for this blue light response, it's most likely controlled by cryptochrome. The pigment within cryptochrome that determines its absorbance is flavin adenine dinucleotide, or FAD. And we can see that it has maximal absorbance here at 450 nanometers and has another peak kind of around 370. So what this means is that this pigment is most likely to absorb those photons and much less likely to absorb photons in this region or way out here. And if we think about what we saw in the last slide with these seedlings, this means that when this pigment absorbs blue light, it converts to the active form. And now this active form actively inhibits the elongation. But this form also has its own unique absorption spectra, which shows a depletion in the blue and an increase in the green. So when this form absorbs photons, it then converts to a third form, which is also inactive. So from this, we can see that blue light turns on the photoreceptor, which inhibits elongation, and then green light converts it again to a third form, which then re-promotes elongation. As I mentioned earlier, a lot of times in horticulture, we want to keep PPFD the same and modulate colors of light. And this provides a dose response for something like blue photons. So this is from 20 years ago out of this lab that applied a dose response of blue light and saw that generally increasing the fraction of blue decreased leaf expansion and decreased dry mass. And this is pretty similar to the results we saw with stem length in a few slides ago. Because blue light decreases leaf area and decreases dry mass like this, and because we expect that green photons will reverse this response, we wanted to ask the question, does green really mean go? So we developed an experiment that used these treatments. You can see a few different fractions of blue here, here, and here. And we had no green, a little bit of green, and a high fraction of green. And we were able to combine these in cucumber, lettuce, and tomato to investigate how these two responses affected plant growth and development. This was recently accepted for publication in the journal Plants. This is Boston Swan. She helped collect a lot of the data for this project. And she's now at Iowa State. In addition to being interested in blue and green interactions, we were also interested in how this might interact with intensity. So I already showed you these low intensity plants here at the top, and these are the high intensity at the bottom. So we can see a few things immediately. First of all, that there's this clear, obvious pigmentation difference between high and low light, but also that in terms of leaf expansion, there was no huge difference between both intensities. and the effect of blue photons was pretty similar between the two. It did turn out that the higher intensity did induce more dry mass accumulation, and they had thicker leaves. So the first parameter we're going to look at here is stem elongation. And similar to what I showed you before with shade avoidance, plants in response to shade will elongate their stems to try to reach out of it. And we can see here that increasing the fraction of blue as expected decreased that stem elongation. And there was an interesting interaction with intensity here in tomato. But now if we look at the response of green, again, they showed the expected response. If green is a signal of shade, we saw an increase in stem elongation in both cucumber and tomato with increasing green photons. This slide concisely summarizes the response of dry mass in this study rather than showing a bunch of graphs. So what we saw is within the 3 species, increasing the fraction of blue consistently decreased dry mass accumulation, while increasing the fraction of green did promote dry mass accumulation in tomato, but did not really benefit either cucumber or lettuce. Although here I do want to point out an interesting interaction we saw in lettuce. When the fraction of blue was low, increasing the fraction of green promoted leaf expansion and dry mass accumulation in lettuce, but at the high intensity of blue, there was a decrease. So there was this interesting interaction between blue and green in the prediction of lettuce, at least. So from an applied perspective, if we're trying to create LED fixtures for best case plant growth, it seems useful to minimize your blue, and maybe in some cases with tomato, and if your blue is low in lettuce, it might be good to increase the fraction of green. Okay, so coming back to this slide, we talked about blue and green responses, and now I want to focus on this far red region. So I don't really know what other people's experience is reading the literature, but when I read through things, basically you read through a really interesting introduction, and then there's also an interesting discussion, and they cite their references in there. So you go back to the older literature, and there's something really interesting to read there. And so you keep going back and further back in the literature until finally you're reading papers from the 1930s and 40s to understand how we originated our thinking on specific topics. I will note that papers from the early 30s and 40s are much more difficult to read than more recent papers. So this is a timeline I made 3 years ago, talking specifically about our development of thought on specifically how the phytochrome photoreceptor perceives light and interconverts between its 2 forms. But I want to zoom in on these 2 guys who I showed you towards the beginning of the presentation. This is Harry Borthwick and Sterling Hendricks, who are 2 of the people credited with the discovery of phytochrome. So I'm going to talk about the experiment they conducted that led to this discovery. If you have some seeds and let them absorb water, trying to get them to germinate, they won't respond if they're placed in the dark. But if you give them a pulse of red light, they'll have nearly 100% germination. But the interesting thing that happens is when you follow this red pulse up with a far red pulse, this brings the percent germination from 100% down to about 50%. And you can actually run this experiment. I read somewhere they did it 100 times. And it doesn't matter how many times you flip-flop this back and forth. The final light that the seeds see determines whether or not they germinate. So if the last light they see is red, they'll have a high rate of germination. If the last light they see is far red, the germination will be really low. So this was conducted in the 1950s. And what they determined was there was this photoreceptor within plants that acted like an on and off switch. Red light turned it on, and far red light turned it off. Eventually, this pigment, as they called it at the time, photoreceptor now, became known as phytochrome, and its 2 forms were called phytochrome red, or PR for short, and phytochrome far red, or PFR for short. So the typical thinking on this now is that PFR is the active form and PR is the inactive form. So red light turns the photoreceptor on and far red turns it off. But this is somewhat of an oversimplified picture of what's actually going on. If we look at the absorbance of this photoreceptor, it is highly sensitive to red and highly sensitive to far red. So the names are fairly apt, but it does absorb across this whole visible region and into the UV. So what this graph shows, it's not exactly absorbance. This graph was originally developed in the 1960s, but then they did a lot of work in the 1980s to really improve the shape of the curve. So what these curves represent is how quickly photons at different wavelengths will convert PR to PFR or PFR back to PR. So you can see that a photon right around 660 nanometer will induce much quicker conversion of PR to PFR compared to a photon at 730 converting PFR back into PR. So if we have a background light spectrum that's constant, eventually these two arrows are going to equal each other, and you'll reach what's called photoequilibrium. And in the case of phytochrome, it's called phytochrome photoequilibrium, and we call this PPE for short. And we can use this equilibrium to determine the relative concentration of both forms of this photoreceptor, the on form and the off form. So basically, what this curve can tell us is the relative concentration of our active form of the photoreceptor. And back in 1976, David Morgan and Harry Smith determined that they could use this parameter of phytochrome photoequilibrium, also called PPE, which again is also the relative concentration of your active photoreceptor. That's what this axis represents. It was a good predictor of stem elongation rate. So this phi sub e and this estimated PPE, these mean the same thing. And now this is a paper from some of our colleagues at Michigan State, Eugen Park and Eric Runkle. And we can see that 40 years later, we're still using this same metric to predict how far red will affect the phytochrome photoreceptor and then modulate plant growth and development. So I reconducted this experiment using different spectral backgrounds. And generally, I got the same data, assuming you just look at one spectral background. So we used high blue, high green, and high red. These were all basically broad white light spectrum. One had a relatively higher portion of blue, one had a higher portion of green, and one had a slightly higher portion of red. And we added different amounts of far red to this, and from the background spectra, calculated this PPE value using those spectral weighting factors. So we found a good correlation for each individual spectral background in PPE with the stem extension rate, very similar to these previous studies. But when we tried to run a single line through all the data, we saw that the correlation was fairly poor. And in thinking about why this might happen, I think I mentioned earlier, but those weighting factors were determined from phytochrome that has been extracted out of plant tissue and into a solution. And then they run flashlights back and forth on it and see how quickly it converts back and forth to determine those weighting factors. But phytochrome is not in solution, it's within plant tissue. And plant tissue has a whole bunch of chlorophyll in it, which can distort the light that occurs within leaves. So these top four images basically are showing how deep light of different wavelengths penetrate into a leaf. You can see that blue and red don't penetrate very far. They're quickly absorbed by chlorophyll, whereas green and far red penetrate really deeply. And this is pretty similar to those curves of measurements underneath a tree where far red is enriched, green is slightly enriched, and blue and red are reduced. So what this curve represents is the average intensity of different photons given the photon intensity above the leaf. So this represents the actual intensity within a leaf. And we can multiply this spectral distortion function by our photoconversion coefficients and develop these new weighting factors. So these red curves represent what phytochrome is actually doing if you extract it into solution. but these green curves are a much better representation of how phytochrome is interconverting within a leaf. So we used our new modified, those green curves, to predict PPE within a leaf. So this top graph is the same graph I showed you before, and the bottom graph is PPE estimated within the leaf. And you can see that, as before, we had a high correlation between any individual background, and we still have a high correlation between any individual background. But now if we run a single line through all the data, the correlation coefficient goes up from 0.47 to 0.75, so it's significantly improved. We submitted this to a journal, and we got reviewer comments back, and the reviewer said, well, you haven't really proven that chlorophyll is distorting how phytochrome is perceiving light, because you still have phytochrome in the tissue. You really need to remove the phytochrome or remove the chlorophyll to really prove what you're trying to show. So can you think of an experiment that is able to really truly test this? So we did some thinking and we came up with an experiment. This kind of stole from some ideas in the 1980s and 1990s, but we applied a herbicide called norflurizin. This is an herbicide that blocks the synthesis of carotenoids. Carotenoids are used for photosynthesis, but they also help block excessive light to chlorophyll, which eventually damages the chlorophyll and breaks them down. So when you apply this norflurizin and put the seedlings under high light, the chlorophyll ends up getting degraded. So we were able to have some seedlings that had chlorophyll and some that didn't. So we again applied our weighting factors to these seedlings and saw how elongation was correlated with estimates of PPE. So these graphs on the left-hand side of the screen are the same two graphs I had already showed you, and these graphs on the right are the new are the seedling studies. So with this seedling study, these filled in circles up here are the seedlings with chlorophyll, and these open circles down here are the seedlings without chlorophyll. And interestingly, although this whole plant measurement still showed a high correlation with each individual background and the estimate of PPE, this estimate or this technique that compared both seedlings with chlorophyll and those without in a single spectral background saw fairly poor correlations. These are low numbers and it was poorly correlated. Whereas our within the leaf estimates showed a high correlation for each spectral background. And in addition to that, we also showed that the correlation through all the data, similar to before, was significantly increased. So basically, this shows that although these top curves are what's really happening with phytochrome as a molecule, when we look at phytochrome within a leaf, these green weighting factors are much more appropriate. So this paper is tentatively accepted in the journal Frontiers in Plant Science. I have 2 positive reviews for it, so I think it's going to move forward. One last thing I want to say about this data is this whole phytochrome photoequilibrium technique is pretty complicated. We're looking at how light affects the transition between 2 states of a photoreceptor and how that then goes on to control growth. And it's a really complicated technique, and we wanted to see if we could develop a much simpler technique. So we developed this percent far red metric, and sometimes we also call it the far red fraction. But basically, this just takes the flux of the far red photons divided by the flux of the total photons and uses that to predict something like the stem elongation rate. And using the same data as we used in the study I was just showing, we see that this is a great correlation through all the data, and there's a good for any single background and all the data together. This idea as a simple environmental metric was published in the Journal for the American Society of Horticultural Science earlier this year. We didn't actually use this data, we used a different set of data, but it showed basically the same thing. Okay, so now if we come back to this slide and we look at the y-axis here, this says relative photon flux. So what I wanted to show here was that how the quality of light changed when you moved into the shade. But when you move into the shade, it's not just the quality that changes, but also your total intensity will also decrease. So not only is this relative increase in far red a signal of shade, but also this decrease in intensity can be a signal of shade. And it's because of this, we wanted to see how these two parameters might interact in controlling plant growth and development. So we conducted a study where we applied a few different fractions of far red with a few different intensities and measured how changes in things like leaf area and stem elongation. We did this both in cucumber and lettuce, and I'll show you the data from cucumber first. There were some interesting interactions, but for the most part, the data is much more interesting in lettuce, so I'll linger on that longer. What you can see is generally as you move in the upward diagonal direction in this image, you get an increase in size of the plant. So increasing far red increases leaf expansion, and increasing intensity increases photosynthetic rate and increases growth. If we look at stem elongation, stems would be expected to elongate the most under the lowest intensity and the highest fraction of far red, which are these plants down here. But if you compare these plants with the higher intensity, we can see that these are actually taller than those. And it is interesting to wonder why that might be, but the answer is fairly obvious when you think that these were grown at extremely low light. Basically, there was just a lot less photosynthesis. They were developing slower, so they ended up being really short. We can compare, though, this intensity effect with elongation between this high intensity and this middle intensity, with this middle intensity being taller plants than the high intensity. Okay, so moving on to lettuce. With cucumber, we generally showed that increasing far red and increasing intensity both increased leaf expansion and increased dry mass accumulation. But if we look at lettuce at the high intensity, increasing the fraction of far red, in this case, increased leaf area and therefore dry mass accumulation. But if we then look at the low intensity, as we go from low to high far red, we actually see a decrease in leaf expansion. And so there's this really interesting interaction between far red and intensity when we look at lettuce. Looking at the literature, it is interesting to see what different people report for the effect of far red on leaf expansion. If you look at a lot of horticultural studies, they will often report that far red expands leaves. Whereas if you look at more physiology studies, they typically show that leaf area is decreased with an increase in far red. So there's a bit of a contradiction in the literature there. But if you look at the intensities, for the most part, from these two studies, horticultural studies are typically done at higher light, and the more basic physiology studies are done at lower light. So this may explain this interaction or this contradiction in the literature. Okay, so here we're just looking at a graphical representation of what I was just showing you in those images. This is leaf area here with increasing percent far red at high intensity, the middle intensity, and low intensity. And what we see is that, similar to the picture, increasing the fraction of far red, increased leaf area at the high intensity, and at the low intensity, increasing the fraction of far red, decreased leaf area. There was a bit of a statistical issue comparing these because we were comparing small numbers with small variance to big numbers with a big variance. So we thought the best way to get around this was to try to normalize the data to the no added far red control. So when everything was normalized to that value, we could see that yes, statistically far red increased leaf area at the high intensity and decreased it at the low intensity. So we wanted to do a little bit more investigation of why this might be occurring, why this interaction between far red and intensity is occurring within the plant. So the first thing we looked at was specific leaf area. Specific leaf area is calculated by taking leaf area and dividing it by leaf dry mass. And it's basically an indication of how much leaves are expanding given the biomass partitioning to the leaves. As specific leaf area increases, you'll have a much thinner leaf that's spread over a wider area. And what we found with the data is that specific leaf area under all three intensities increased with increasing far red. Again, when we normalized it to the no added far red control, we saw no interaction between intensity and far red. So this to us indicated that far red was not changing leaf expansion at these different intensities, and something else was kind of controlling that total leaf area response. And as I mentioned, specific leaf area is the indication of how leaves are expanding given the allocation of biomass to the leaves. So we wanted to look at how plants were allocating their biomass between different organs. So if you look at increasing percent far red here at the high intensity, we see that there was no effect. The lettuce plants allocated very little biomass to their stems when the intensity was high. But now if we compare it to low intensity, as we increase percent far red from 2 to 33%, we see that the lettuce plants allocated much more biomass from their leaves to their stems. And basically this means that the total leaf area response that we were observing was caused by biomass being taken away from the leaves and moved into the stems. So I also put stem length on this graph to just show that these two responses are closely related to each other. And they probably both kind of drive each other. Stem length is controlled by how much biomass is partitioned and vice versa. Okay, so moving on to the last study now. I think I've got a little bit of time. I made another timeline where this was for a seminar last semester where I started working on flowering control. So I made this timeline of how our thought has developed over the years on how light controls flowering. So a lot of these people in the early stages of discoveries are very similar, but people in the later ages are different. One person I want to point to on this graph is Frank Salisbury. Frank Salisbury played a role in understanding how the circadian clock within plants was set. So all plants and animals both have this internal circadian clock that tell them when it's day and when it's night. So for humans, it helps us start to get sleepy at nighttime and wake up in the morning. And similarly, the circadian clock plays a role in flowering. So Frank Salisbury's work helped elucidate how this clock was set. And a lot of his work found that it was really the morning or dawn signal that set the clock. Now that's not as important. More important about Frank Salisbury is the people he's associated with. So here I have Ray Wheeler circled here right behind Frank Salisbury. And Ray Wheeler's on my committee, he's here today. And then this here is a young Bruce Bugbee, my advisor in the 1980s. Okay, so this is something that has nothing to do with anything I've published, but it's really cool and I'm proud of it, so I wanted to show it. So as I mentioned in the last slide, plants have this circadian rhythm, and then they, which controls flowering. But one of the first instances of how we knew plants had this circadian rhythm was looking at leaf movement. So for some species, during the day, they have horizontal leaves, and at night, their leaves turn vertical. So their leaves move up and down with day-night patterns. And if you move these plants from this daylight oscillation all the way into the dark, they'll continue moving up and down for a few days. So this response has been known about for, I think, centuries now. And I thought for sure that there would be a YouTube video showing it, but I couldn't find one. So I had my lab buy a camera so I could test it myself. So when the leaves are dark like this, that means it's daytime. And when it is, when they're kind of white colored like this, that means it's nighttime. So you can see that this plant is opening its leaves during the day, and at night it closes the leaves back up. This is a mimosa plant. We set this to a dawn at 6:30 a.m. and night at 6:30 p.m. And this was the last day I gave it light. So now I moved it into constant darkness. And we can see that when the clock hits about 6:30 a.m., the leaves open back up. And when it becomes nighttime again, when they think it should be night, the leaves close. And in the darkness, the plant continues this oscillation for about 2 days. But after that, with no input into the circadian clock, the signal starts to dampen out a little bit. And in fact, by the end of this video, it should be nighttime and some of the leaves are still open. So it's lost this ability to control timing. Okay, so now on to what the last study actually is. So the camera we used to take that night vision video in the last slide uses a near-infrared camera. And this has a tail way out into the far red region. And we wanted to investigate whether or not this could potentially have effects on plant development, especially since they're used in security cameras. And there was a good reason to think it could especially affect flowering, because if we look at this tail for the activation of phytochrome, it shows some sensitivity between 700 and 800 nanometers. And there's some overlap between the NIR LED and the curve for the activation of phytochrome. So here it's important to note that there are some species that can be inhibited from flowering if they're given light during when it should be dark. And two of the species that we decided to look at were cannabis and soybean. So one of the people who did a lot of work on flowering in the 70s and 80s was a scientist named Daphne Vince-Prue. And she wrote a few books on flowering. And in one of her books, she lists the sensitivity of different species to night pollution lighting for inhibiting flowering. And soybean and cannabis are two of the most sensitive species she lists in that table. So here's the design of the study. I think you can see it here. If you look at the very top of this image, there's two very faint red lines. And if we brighten everything up, we can kind of see these are the NIR LEDs I applied for a whole 24-hour period, while the daylight was on for only 12 hours. And the intensity that we applied this NIR at was actually brighter than like office lighting, but because our eyes are so insensitive to it, it appears barely visible to our eyes. So what we ended up seeing was that, yes, the NIR could affect flowering. Basically, it increased time to flowering as we increased the dose of NIR photons. Increasing time to flowering means flowering was delayed in both these species, meaning that these NIR photons were enough to activate the phytochrome photoreceptor and inhibit flowering. So this paper is still in review. Two of my co-authors on it are Mitch Westmoreland and Shuyang Zhen. Hopefully it gets accepted, but we'll see what happens. Now there are a lot of people I'd like to thank. It's been a lot of help over the last 5 years, and I will open it up to any questions.
I need the microphone to make comments.
I think, sure.
I just want to make one comment. Over the 5 years that Paul's been working on this, it occurred to us in the lab that our mantra for the lab is something like, and I think we'll put this on t-shirts. Sure, it works in practice, but does it work in theory? And that's what Paul's been getting into for multiple years. His parents visited here a couple of years ago, and I could see he came from a deeply research-oriented family. So he got his start well before he came here. But we'd be happy to take questions from the audience. And then after we're done with that, we'll excuse the audience and the committee will ask further questions. So Paul, I'll give you the microphone back and any questions from, you can come back in here from the audience, or maybe even general questions from the committee.
Paul, this is Mark. I can start it off since everybody else seems to be shy. Related to your last piece, has anyone ever looked at the phytochrome absorption past 800 nanometers?
You asked about the absorption of phytochrome past 800?
Yes.
I've looked through the literature and I've tried to see the absorption of phytochrome, someone publishing on it, and I can't find anyone. It seems that 800 as far as anyone has gone. It may be due to just the instruments, the fact that they just measured up to 800 and stopped there. I would guess that it's, you know, almost logarithmically exponential decrease as you go further and further into the from far red into the near infrared region. So it's definitely hard to say, and I would guess that if you were to take a photon at 805 nanometers and apply a really high dose, and it was a laser, so extremely narrow, I think you could potentially have some effects on growth, but you would have to apply a really high dose. And then by the time, I would guess eventually the photoreceptor just isn't going to absorb anymore, and you won't have a response.
But it's not uncommon for pigments to have multiple peaks, right? So to me, it seems like a very simple alternative explanation for your data would be the potential of additional absorptance peaks in that part of the spectrum.
So you're suggesting that there might be another peak beyond 850 or 800?
Well, nobody seems to have provided any evidence that there isn't, right? So I don't think you have any data that rules that out. Let's put it that way.
Yeah, sure, that is true. It's certainly possible. It's also interesting though that if you take a general leaf absorption spectra, it generally shows minimal absorption between 800 and 1,000 or so nanometers. And phytochrome is in low concentrations in leaves. So it's possible that if there was this peak, we just wouldn't be able to pick it up in the absorption spectra. So it is possible that there is another peak out there. I'm not sure it's likely, but it is possible.
Yeah, so I would imagine that if you had a phytochrome deficient plant, deficient in all forms of phytochrome, and you would measure the leaf absorption spectrum, that you wouldn't see any impact of the absence of phytochrome. But that's just pure speculation on my part.
I think that's probably true too, yes.
Thanks.
Hi, Paul. It's Ian Ashdown here. I've been doing similar experiments with the 850 nanometers as we've been discussing. With my system, I already had a decent far red ratio, 725 nanometer LEDs in the spectrum. The effect on basal was about 5 times the stem elongation when I had the 850 nanometers on it. So I'm almost in agreement with Mark that I would suspect there may be another spectral absorption peak beyond it, because there's absolutely no emissions from the LEDs below 800 nanometers in my experiments. You mentioned, by the way, somebody, Daphne, which you had a paper. I'd be interested if seen that paper. Do you have the full name of that woman again?
Her name is Daphne Vince-Prue. I can send you the book chapter where she lists the relative sensitivity of night pollution of different species. It's in a book called Photoperiodism in Plants. Yes. So your response you observed in basal, it is sort of the opposite response we're seeing with flowering. With flowering, we're looking at the activation of phytochrome, which actively inhibits flowering in both of these short day species. But with elongation, what we're seeing inactivation of phytochrome, which induces elongation. But did you say you saw 5 times the elongation?
Roughly, yes.
Okay, that is fairly hard to explain with just a small amount of NIR and just looking at how that tail affects the inactivation of phytochrome.
Yeah, that's what I think. I can't explain it. It's shade avoidance, but it's quite spectacular, the effect. I will note that I've got a colleague at Beloya who ran the same type of experiment with cannabis, and he saw absolutely no effect on it, on the seedlings. So it's definitely species specific.
One thing we haven't been looking at with the NRR2 is maybe there are some subtle changes in leaf temperature that are causing some effects. That might be interesting to look at as well if this increase in the flux of NIR is heating up the leaf, and that might be having some effect.
True. He ran 1400 nanometers as well and saw no effect for those LEDs. So that more or less ruled out any infrared heating as he saw it. More research required.
Hey, Paul, Matt here, Matt Hardy.
What's going on, Matt?
I really enjoyed your presentation. Thank you for sharing it and inviting me. In one of your early slides, you basically showed that with a higher proportion of blue light at a particular intensity, that it would decrease biomass production.
Yes.
So how low of a proportion of blue can you go before you start to have like adverse effects? And maybe I should wait until you get there, but yeah, especially considering that blue light is high energy and possibly more expensive to provide.
It's a great question. I originally was going back to the original slide, but based on your question, I decided to move forward to this slide. This is actually something we've been working on recently. So it's brand new data, but especially from this idea of, if you look at LEDs, efficiency isn't actually the right term. Efficacy is the more important term. Photons drive photoreactions. And so because red photons are lower energy than blue photons, if you can have a higher portion of red photons in your spectrum, your system will be more efficient. And because of that, we want to ask the question of how low of blue we can go. So we decided to test that in some kind of preliminary studies. And this is Rex lettuce grown with 10, 5, 2.5, 1, and 0% blue. And we wanted to see basically, can we grow these plants under pure red and not get adverse effects? The literature is somewhat inconsistent on this. Sometimes it shows that, it's no problem, and actually you can increase dry mass by removing blue. But other studies show that you pay a big penalty by removing blue entirely. You can notice some subtle differences between these. Early on in this Rex lettuce, especially, I don't have photos of it, but these older leaves you can see were really elongated. And this plant looked kind of crazy at the beginning, just really long, thin leaves. But it seemed to have grown out of it. And so it's interesting to think about how the age of the plant affected this response. But I think it's very species dependent on whether or not you could remove blue entirely. I think for some species, you're okay doing it. Others, maybe not so much. It also depends on the applied perspective. If we're just interested in dry mass accumulation, then that might be good to entirely remove blue. But often, especially with lettuce, if you remove blue, even if you get an increase in dry mass accumulation, the lettuce leaves look elongated and stressed and not really pleasant from a marketable point of view, which isn't very good. So in that case, you definitely wouldn't want to remove it.
Do you know anything about like the nutritional content? Do you know if anything like that would be affected, or have you just looked at like the biomass?
I'm generally fairly skeptical. If we're talking about nutritional content in terms of like nitrogen, phosphorus, things like that, there are some reasons to think that light will have an effect on nutrient uptake. And a lot of studies report on how light affects nutrient uptake. It's not something I've looked into a whole lot, and I'm fairly skeptical because I haven't looked into it as much that the responses are kind of driven by photoreceptors. If we're talking about nutrition in terms of carotenoids and anthocyanins, this actually brings up another skepticism of, we often say carotenoids and anthocyanins are good for human health. Whether or not they really show demonstrable benefits, I think is another thing. But definitely as you increase the fraction of blue, you tend to increase accumulation of flavonoids, carotenoids, anthocyanins, which are all beneficial components as antioxidants potentially. And there are physiological reasons why the light would increase those components. So you can benefit, if these compounds are beneficial, you can potentially increase them by increasing the fraction of blue.
Okay, nice. Thank you.
You want to make a comment?
Okay, I'm looking at the people on this, and Kevin Folta is on this. Paul's old mentor from Kevin's, not that you're old, Kevin, but Kevin's early mentor from University of Florida. Ep Havelink from Wageningen is on the call. Ian Ashton just asked a question, and Ian, I think you're in England, aren't you? So we're all over the place with this. Yeah. So we got some a lot of luminaries, and Paul's family is on this call as well. So it's nice to be able to, one of the good things about COVID is we get better at doing these kinds of things, and people can join from all over the world. Back to Paul.
Okay.
Kevin, you haven't asked any questions yet. Oh,
Oh, you're muted, Kevin.
Probably a good thing. I just said thank you for the invitation. So I don't have any questions. It looks really nice.
All right, thanks, Kevin. Okay,
okay.
I'll give it back to you again.
This might pick me up anyway. So at this point, now, by the way, I think this is recorded too. Yeah. So if there's other people that might be interested, it's we'll make sure we've got the link posted here, and I need to be in the middle, and other people can see it. So at this point, we'll excuse all these people except the committee members who will have continue with further questions on this. Thanks for attending. And just once again, being on a committee is a great service to academia. And in this case, just to introduce Paul's committee again before the other people leave: Mark Van Eersel, University of Georgia; Ray Wheeler at NASA Kennedy; and Lance Seefeld right here from our chemistry and biochemistry department. And then finally, Professor Larry Hips from our plant soils and climate department. Thanks for tuning in, and we hope we can stay in touch with all of you in the years to come.